A typical antibody drug is made up of approximately 1,300 amino acids, synthesized, folded, and covalently bound together into a 4-chain, 150 kilodalton structure. This assembly is carried out entirely by living cells, which means there is inherent variation even within single batches of an innovator's product. This variability is a major challenge for the developers of a biosimilar drug. Biosimilar developers must thoroughly understand the structure and variation of the innovator product to set specifications for their biosimilar. Any structural differences must be characterized and evaluated for biological significance. Liquid chromatography mass spectrometry (LC-MS) methods allow us to measure and compare key attributes such as intact mass, amino acid sequence, post-translational modifications, disulfide linkages, glycan profile, and higher order structure. Intact mass analysis is the cornerstone for understanding the structure and variability of large molecules. This spectrum shows a range of molecular variants present in a single sample of the innovator product. Each heavy chain contains one N-glycosylation site, populated by a range of glycan structures. LC-MS allows us to make quantitative comparisons of the relative abundance of major variants. Intact mass analysis allows us to establish a basic level of similarity and may highlight possible differences for further study. A protein is constructed from a linear sequence of amino acids. That sequence is specified by the genetic code, but the potential for errors in cloning, expression, or in the reference sequence means that the amino acid sequence of innovator and biosimilar must be verified. In peptide mapping, proteases break the protein chain at predictable sites. UPLC-MS data from a protease digest can confirm the presence of peptides of expected mass. MS to the E fragmentation analysis can confirm the sequence of individual peptides. Results from different proteases are combined to ensure 100% coverage of the sequence. This process of confirming the order of every peptide bond is a requirement to show sequence similarity. Sites within proteins are susceptible to modification during and after production. For example, antibodies are particularly susceptible to chemical oxidation of methionine and tryptophan and deamidation of asparagines and glutamines. The mass change from oxidation is readily observable by LC-MS of a protease digest. Here we see a peak indicative of a peptide with oxidized methionine. Relative peak area reveals differences in the presence or amount of oxidation. MS to the E analysis can pinpoint the oxidation site. High-resolution MS allows us to detect the single Dalton delta of deamidation. The penny K peptide is a common site of deamidation. Here we observe similar low levels in innovator and biosimilar samples. These lower abundance species often coalute with other peptides, and the selectivity of MS is required to detect and quantify them. The folded structure of an antibody is stabilized by internal disulfide linkages within the subunits, between the light and heavy chains, and holding the heavy chains to each other. These disulfides can be misformed or scrambled, leading to misfolded product variants. The pattern of disulfide linkage can be examined by performing a protease digest under non-reducing conditions. LC-MS of the digest reveals the presence of peptides still joined by their disulfide bonds. Purpose-built software can identify canonical and mispaired disulfide linkages. One of the most challenging aspects of creating a biosimilar is optimizing a production process that yields a glycan profile consistent with that of an innovator molecule. While intact mass analysis can provide an overall glyco profile, assessment of critical minor glycoforms requires more detailed analysis. N-glycans can be released by digestion with the enzyme PNGase-F and labeled with a reagent such as Rapiflor-MS. UPLC fluorescence MS of the labeled glycans gives a more accurate picture of the major and minor glycans present. In data from an innovator and an approved biosimilar, we observe the consistency of glycoforms, with acceptable differences in the relative levels within the profile. The generation of high-quality glycan profiling data will demonstrate to regulators the comparability of key attributes of drug structure and your ability to control the production process. The primary structure of a protein, determined by the sequence of its amino acids, is folded during synthesis into sheets, loops, and helices that provide a secondary level of organization. The higher-order interaction of these local structures forms larger structural domains, and the assembly of multiple folded chains constitutes the quaternary structure of the molecule. These multiple levels of folding are critical to the function of the protein. Hydrogen-deuterium exchange with mass spectrometry, HDX-MS, is a powerful method for assessing and comparing higher-order structure. If the protein is exposed to heavy water, it will begin to accumulate deuterium. The hydrogens on backbone amide groups exchange with deuterium at a rate that depends on solvent accessibility and involvement in hydrogen bonding. With careful control of experimental conditions, this allows reliable and reproducible study of conformational dynamics. Acid protease digestion allows us to monitor deuterium uptake at a more local level on individual peptides and assess the degree of exchange. A time course of increasing deuterium exposure allows us to calculate an incorporation curve for each peptide. This technique allows us to quantify structural differences between samples. Closer inspection of the data enables us to observe a minor structural difference between innovator and biosimilar. We can map these differences on the complete structure. This peptide's location suggests glycosylation may be responsible for the differences observed. HDX-MS gives us reliable insight into higher-order structure and dynamics of biomolecules and can reveal functional differences not apparent from primary structure analysis. Liquid chromatography with mass spectrometry is an essential tool for determining primary and higher-order structural similarity between an innovator and a biosimilar. The combination of protein level, peptide level, and higher-order structure studies empowers a biosimilar producer to systematically demonstrate comparability of key product attributes. Your ability to execute these studies will form the foundation for successful regulatory submissions that establish biosimilarity to an innovator molecule. Waters, the science of what's possible.